Hello, everybody. It's 11 o'clock and it's time for a very short welcome on behalf of the Danish-Swedish Life Science Cluster Organization, Medicon Valley Alliance. My name is David Muniz-Sabernik. I'm the Head of Business Development and Public Affairs with MVA, as we call it. I hope and truly believe that you will enjoy today's inspirational webinar with the title Molecular Classification-Based Identification of Diagnostic and Treatment Targets in Glioma, co-organized with our member company, GenScript. If you get inspired not only by the concept of today's webinar, but also by the concept and start to wonder if it would make sense for your company or your organization to organize an inspirational webinar with Medicon Valley Alliance, feel free to reach out to me. You will find my contact info on our webpage, and it will also appear on the final goodbye slide after the presentation. If you have questions about Medicon Valley Alliance membership, how to join the MBA networks, which, by the way, employees of MBA member companies can join free of charge, give me a call or send me an email. I also strongly encourage you to visit our webpage event calendar, where you will find a number of hopefully interesting online and offline events organized either by Medicon Valley Alliance or by some of our member companies and partner organizations. Next slide, please. Just a few practical remarks. Our household rules for today. It's pretty much standard operating procedure, but as you can see, you are automatically muted. You should use the chat for technical questions only, and then use the Q&A for questions to our speakers, and be aware that this event will be recorded. You will receive an email afterwards with a link to the recordings. Enough said on my part. I am not the reason why you tuned in today, so let's fasten our seatbelts and get started. And without further ado, over to Nicolas Gao, who will be your moderator today and who will introduce Dr. Fan, who is, of course, the real reason why you were tuned in today. Nicolas, the floor, or in this case, the screen, is yours.
So good day, everyone. Thank you very much to David and the Medicon Valley Alliance for helping to facilitate this webinar today. My name is Nicolas Gao, Marketing Specialist at GenScript Biotech Europe, and I'll be the moderator for today's webinar on molecular classification-based identification of diagnostic and treatment targets in glioma, given by Dr. Xiaolong Fan. Speaking of Dr. Fan, Dr. Xiaolong Fan is a professor of neuroscience at Beijing Normal University, having completed his PhD in human genetics and his postdoctorates in bacterial infection biology at the University of Greifswald in Germany between 1989 and 1997. He then moved on to the Faculty of Medicine at Lund University in Sweden, where he worked on hematopoietic stem cell biology and glioma biology, and became a principal investigator there in 2001. Dr. Fan was then recruited to the Beijing Normal University in 2009, where he is developing a bioinformatics platform that seeks to integrate big data analysis with pathways of organ development and cancer pathogenesis. It is also where his lab developed an EGFR module / PDGFRA module classification scheme for glioma identification, as well as the MCL-1 classification scheme for multiple myeloma identification. Dr. Fan has also been recognized by the German Paul Ehrlich Society for his work on researching the roles of lipopolysaccharide binding proteins in the innate immune system response to gram-negative bacteria. And we here at GenScript Biotech Europe are honored to be supporting his research and look forward to continuing to support Dr. Fan in his future research endeavors. In this webinar, Dr. Fan will be providing a high-level overview of his 15 years of experience dealing with the challenges posed by his research project, as well as outline what his and his lab's plans are for the future. I would now like to welcome Dr. Xiaolong Fan to begin his presentation.
Can I start the video? I cannot start my video. Okay, now it's, can you hear me? I hope you can hear me clear. You can hear me. If you can't hear me, you should raise your voice.
Yes, we can hear you.
Okay, thanks. That's great. So because we're far away from each other, I'm actually now in Beijing under quarantine in a hotel. So I'm very happy that to have this possibility to introduce what we are doing. So we work on. I'm from Beijing Normal University. Here is my contact information. So we work on molecular classification of adult glioma. So this is a project which we have developed since around 2008. So I have continued when I started here in Beijing, and I think that the most important issue is not the data itself, but the concept. How we want to do this, and some results. What we have generated with the results. So instead of working on individual genes, we actually tried to work on gene network to do disease classification. I think that what we found for glioma is also applicable for other diseases. Here is a summary of what we found actually. So we use different gene networks. Here you see if this network is high, so indicates one subtype. If another network is high, indicates a second subtype. That's vice versa. So, sorry, I'm not sure. Okay, let's see. So before I go to the details, we should I present the problems, the challenges in the field. So this is a classical outcome, so or the persistent outcome for in the glioma field. So we have many publications, many projects, but despite this, we have only one standard treatment. The outcome is like this. So we cannot, or we can hardly improve the outcome for the patients. So far, all the phase 3 clinical trials for the most aggressive form, also the most frequent form of human glioma have failed. We have no success here at all. So we do not know what to do. That means actually we don't have a target. Though we have many papers, we have many publications, many projects, but we have not been able to identify actually one single useful target. How could this happen? So we look at the other field. If we can look at the other field, this is probably the best successful example in the treatment of cancer. This is in the childhood leukemia, B-cell acute lymphoblastic leukemia. You see, nowadays, so over 90% of those child, if they have this leukemia, they will survive over 10 years. But 60 years ago, the situation is quite similar to glioma. So what happens with, so what's success here? So we look at it. The first issue if we are able to treat cancer is that the proper diagnosis. Here, the diagnosis for the leukemia is probably the key for the advanced cell, both in terms of etiology study and also the clinical advances. So with the normal hematopoietic as reference, when we have leukemia, we will diagnose leukemia into myeloid or lymphoid lineage, and within each lineage, we will define the stage of differential blockage, and then we will also define the pathways of differential blockage. So the childhood leukemia has a blockage at this stage. So imagine what would happen if we mix this up. We mix myeloid leukemia and lymphoid leukemia up. So we can still recognize it as cancer, but we will not be able to find the clue why there is a cancer, how to treat them. That's the problem. And so we can look at it, how glioma is diagnosed. So nowadays we have the so-called integrated histomolecular classification scheme. Here we have histology, we have also genetic events. So we call this integrated histology and molecular classification scheme. We are good at distinguish normal and versus tumor. We can also somehow predict the prognosis, but we actually have not been able to find a clue how glioma occurs. So the principle, why this is a problem with this classification or the diagnostic scheme. So it's used worldwide as standard, but it's really a problem. Why it's a problem? Simply because we assume there is an astrocytoma, there is an oligodendroglioma, there is the glioblastoma. And this is a text. I'm sorry, this Zoom software does not allow me to read this text. But it's you can, if you read the original text from the 2016 WHO classification guideline, it says that you presume glioma would originate from astrocytes, from oligodendrocytes, from stem cells, and then you make this, because some gliomas show morphological similarities to astrocytes, oligodendrocytes, and here we do not know which cell is similar to this type of glioma that we call glioblastoma. This probably come from stem cells, and we can look at the classical paper how the morphology occurs. So this is a classical example published many years ago. So we have a fibroblast. We introduce raw, RAC, and CDC42. You can see suddenly those cells show different faces. So morphology itself will not be able to tell you the cells of origin and the stage of differentiation. So based on morphology-based diagnosis, it's good to distinguish tumor and normal, but it's very bad to guide us for etiology studies. And so this principle has been taken into the newest classification scheme. So, but the system is now much more simple. So we can have a wild type glioma, and we can have IDH wild type glioma. We can have IDH mutant glioma. So IDH mutant glioma is further divided into astrocytoma or oligodendroglioma. So oligodendroglioma have 1p19q co-deletion, astrocytoma do not. So this is the standard, but this standard has a major weakness. The following question. I hope you can see this. The third question I can't see by myself because it's blocked by this Zoom software. So it's a cell lineage issue. Is a cell lineage issue, and is there a differentiation blockage within the lineage? And the third is that I think most important is we define glioma into 3 subtypes. Are all 3 subtypes really distinct biological entity or not? If they are distinct, then should have a distinct etiology. If they're not, then we make trouble for ourselves. Though we know that, though we can know there's a cancer, but we do not know which type of cancer they exactly is. So what we want to do is that we want to mirror glioma into neurodevelopment. Many literatures have indicated glioma would originate from, some glioma originate from stem cells, some glioma originate from from OPC, so oligodendrocyte progenitor cells. They express PDG receptor A, SOX2, and G2. But how can we have a practical approach? We know morphological approach will not be able to do this. How can we have a practical approach to distinguish or to identify which glioma really resembles stem cells, but the other glioma is similar to OPC? This was not possible before. So what we want to develop is that we want to use transcriptome transcriptome-based molecular classification because we believe transcriptome integrates features of cells origin, genomic alterations, and epigenetic changes. But the transcriptome, if we make a simple example, is like that. So it's almost like we look into the sky. So we have a lot of data, and this data, not all data are useful. Some data are just noise. How can we distinguish between noise and signal and find the simple but powerful rules? And this is what we want to do. And how we work on it. So before we do it, we want to introduce a dogma in the field. Here, the people would believe that all the information, just before, all the information is useful. And so unbiased big data analysis has been dominating the field. So here is a paper which published by the very dominant consortium, TCGA, and they clustered gliomas into 4 subtypes. And this paper has been published in 2010, and since then this has been dogma for the field. So the major limitation is that so clinically, you see, different subtype has no difference in the prognosis, and biologically, this say although this it's oversight is cited by 20 almost 2,000 times, but still among those papers, this we do not know each subtype, the pathology. So do they have unique cells origin, unique genetic origin? We do not know. So this is the major problem for the field. So we are guided, we're almost led by the dogma of unbiased big data analysis. We believe all data are useful. So the consequence of that is that this is a science paper published a few years ago. So within each glioma, you will be able to find different subtypes. And further, they will show that because glioma is constantly progressing, and in the same patient, so you will see a random shift between different subtypes. We call it, this is called plasticity. So actually, we cannot find a loose based on this. No loose could be found because it's constantly changing. So that's I already said that. So it's clinically we can't apply this to the clinic. Secondly, each subtype we have a lot of signal, we have a lot of paper, but we do not know which target is specific to each subtype. And we think, so we formulate this kind of concept. So molecular classification for glioma, this may also be applicable for other cancers. We think that the classification scheme should be clinically meaningful, should be not totally new, it should be integrated or anchored in a known pathogenesis. And you can't claim that everything is totally new, the other things is just bullshit. It's irrelevant. But it's always be integrated with existing knowledge and should be applicable in the clinics and should guide, most important, I think, it should guide target identification. So how we did it? So we learn from leukemia diagnosis. We want to identify the lineage, stage of differentiation blockage, and the pathways responsible for the differential blockage. And we hope with this principle, we will be able to establish subtypes. Each subtype will be driven by unique pathogenesis. And therefore, each subtype will potentially benefit from subtype-specific treatment. So we have this hypothesis. We test hypothesis that the convergence between gliomogenesis and brain development would enable glioma subtyping. And we have actually tested test this hypothesis. We have this in mind for a long period, and we came up with EGFR and PDGF receptor A, because many years ago this two molecular was very hot. There were many papers. So also because they are easy to identify, the frequently mutated, amplified in glioma, and it's also important for the stem cell compartment, for the early oligodendrocyte progenitor compartment. And the second approach we used is that we instead of using working on individual genes, we aim to work on gene network, because we believe that so if the genes are involved in the same pathway, they will be co-expressed within the same cell. So within a tissue, if you have multiple cell types, the genes from the same cell type will be co-expressed also. So this is what so the two principles which we have found quite useful, or the other way said it's quite powerful in establishing classification scheme, and probably also really also powerful in identifying targets. So how we did it? So we use a Pearson correlation coefficient analysis with EGFR and PDGF receptor A as the seed gene. I'm sorry, here should be 29, not 19. Should be 29, and here is 40. So we found 29 genes co-expressed with EGFR. This is called EGFR model. We call this EM gene. We found 40 genes which co-express with PDGF receptor A. So here we have the genes which are crucial for astrogenesis. Here we have the genes which are crucial for oligodendrocyte genesis. So the amazing part is that to that time when we found this, we read many papers, but when we did this analysis, we see that all those genes which involved in oligodendrocyte differentiation, they are co-expressed actually, and those genes which are important for astrocytogenesis, they are co-expressed. That was for us was amazing, and we think that was a nature-made beauty. We use mathematics to manifest, to find this nature-made beauty, and so based on this, we have developed the EMPM classification scheme, and this was so we had really difficulties to get this paper published after 2 years finally. So we have worked with many journals. Most journals they think that our approach is not meaningful, and because we were challenging the TCGA consortium, and they were constantly reviewing our paper, and at the end we could convince PNS to take it, and we actually, so our goal was to define glioma molecular subtypes according to cells of origin. So now we will start to explain so the schemes we have formed. So we want to see how is the clinical significance? Is this clinically meaningful? If something is not clinically meaningful, then we can have this in the lab as much as possible. Then it's no use at all. So we have actually analyzed the data from China, from Europe, from America. Now we have more data. Actually, in all these data sets, no matter from which country, from which team, which is generated, or from which platform it was generated, we always see consistent data. And this is like this. So I will first introduce you how this is done. So you see this is clustering results. So on top we have different samples, and here we have different genes. So you will see across different samples, so the PM gene will be co-expressed. EM gene will also be co-expressed. And if a sample have enriched expression of PM genes, it will have low expression of EM genes. And if sample have high EM gene expression, we have low PM gene expression. So based on this reciprocal expression pattern, we are able to distinguish gliomas which show high expression of EGFR module. So we call this EM glioma, high expression of PDGFR module, which we call this PM glioma. So each subtype contains almost all morphological subtype. So the morphological subtype is this subtype is independent of morphological subtype. And we also see this subset of samples, which also has different gliomas, different types of gliomas. But also, I'm sorry, this lower part is blocked here in my screen. I hope you can see it. At this part, we can see the normal sample. So this sample, this is a normal, so non-tumor sample and the tumor samples with a high contamination of a normal tissue. So we can see this is from data from Beijing. We can see that there's so EM samples have poor prognosis and PM samples have better prognosis, and EM low, PM low are those samples mostly actually from PM samples, but it contains lot of tissue of normal brain tissue, non-tumor brain tissue. So the EM PM signature is hidden in this group of samples. And similarly, we can find the clinical outcome from Holland. For the data from Holland, you can see that the EM sample have poor prognosis, PM sample have much better prognosis. And this is similar is from the data from America. So the EM have poor prognosis, PM have better prognosis. So compared with the morphological diagnosis, we can see that for this glioma, this knowledge is a little bit old. We have published this data in 2014. So you will see, but still this still stands. So you see lower grade glioma. We can't, based on morphological diagnosis, we cannot distinguish which one is a better prognostic one, which one is a poor prognostic one. We can indeed distinguish really better prognostic one, poor prognostic ones, according to our classification scheme. And similarly, for the high-grade glioma, we can do the same thing. And so this is the clinical effect. And also another effect, we think this is really important. If the subtype is constantly changing, then we will not be able to find the roots. And we see that from same patient, from primary glioma, primary sample and recurrent sample from same patient. If primary is a sample is EM subtype, recurrent sample is also EM subtype. If the primary sample is a PM subtype, the current sample is also PM subtype. So the EM will progress much rapidly, PM will progress much slower. And this is also true for if we look at the multiple regions within the same glioma. It's not that this region is EM, next region is PM, another region is EM again. It will be either be always EM, always PM or EM. There's no change at all. So it's maintained. We also did a single cell analysis. It's also like that. So within the same glioma, no matter single cell level or at the multiple region level, you will always just one subtype. And we will see whether this is integrated with the existing knowledge of glioma. So we see that the EM PM co-expression is evolutionarily conserved, and this is a mouse brain development data. Here you see the EM signature is in the early astrocyte. PM signature is in the OPC. So this is already co-expressed in the mouse development, is evolutionary conserved. The second is that the region, the EMPM region in glioma genome is frequently amplified or lost. And this amplification and loss cause gene dosage dependent expression. And this is we'll only see like this using our classification scheme. If you don't use our classification scheme, and this looks random, quite random. And so if this feature for the EMPM classification do not exist for the TCG classifiers. Here they have 210 genes for each subtype. Actually, this is purely based on differential gene expression, and most classifiers is not associated with normal brain development, nor associated with genomic alteration. So on the lineage level, so we have a defined gene list which represents developing astrocyte, developing oligodendrocyte from mouse development dataset. And then we apply this gene list to the EM or PM gene list. So we will see that this developing astrocyte is enriched in EM glioma, developing oligodendrocyte is enriched in PM glioma. And then we also look at the differential expression of the stem cell genes. So this is the subventricular zone. In the layer 3, you have the gene module called M13C, and this is well defined by a group in 2008. So this M13C represents the signature of stem cells. So we will see that the M13C is highly expressed in the EM gliomas. And at genetic level, we can see that actually, so you will see, I think you are impressed by this picture. So if it's EM and it's PM, all the PM gliomas have IDH mutation. Nearly no have EM have IDH mutation, but all the EM have 7 chromosome 7 gain and 10 loss. And here we can, under this, we got 2 subtypes. One subtype have 1p19q codeletion, another subtype have TP53 mutation and ATRX mutation. So this is also almost like it's also really black and white. It's you have it or not have it. It's qualitative, it's not quantitative. So it's not here 20%, another one 40%, but it's a yes or no, and yes or no event. So we will conclude like that. So we will conclude this. So the EM subtype is similar to neural stem cells, and PM subtype similar to OPC. And next, we want to say that our principle is applicable in clinics. So we have worked with RT-PCR to assess the EM-PM expression module in single samples, individual samples. And we can see that at the RT-PCR platform, the EM-PM genes are also co-expressed. And then we worked with samples from Sweden and samples from Beijing. We can see that again, so EM sample and PM sample contain all kinds of morphological diagnosis. And if it's diagnosed as EM, it will progress rapidly. If it's diagnosed as PM, it will progress slower. And the age is also different. So the PM patients are younger compared to the EM patient. And we can see that the EM-PM glioma are distinct at the genetic level. Here you see chromosome 7 gain, 10 loss, 7 gain, 10 loss. Here you see 1p19q codeletion. And So next part is that we want to show that we really want to show that the NPM classification can guide target identification, and this is a active field we're working on. We actually I have a limitation that to show you the details because we're submitting manuscript and also applying for some patent, but I could so the principle is like that. So we identified IDH mutant, irrespective of the astrocytoma or oligodendroglioma, PM subtype. So the PM subtype is similar to OPC in the oligodendrocyte lineage. So with this, we're working on like similar scheme like in the leukemia. We want to find the differentiation blockages in this IDH mutant glioma. So this I hope this is so within in the next time, if I'm still invited to give this talk, so we will be able to deliver the full details how we have done it, what's the outcome. The next question. So after I have go through the principles, the concepts we applied to glioma, and you would naturally ask the question: is this principle applicable to other diseases? The answer is yes. We have, using the similar principle, we have established a principle for multiple myeloma classification. And here the principle will be the same, but the gene list, the classifier will be totally different. So the myeloma, so multiple myeloma is not that infrequent. It's the second most common malignancy in the hematologic clinic. So, but this is so looks like a bone disease. So because the myeloma cells sits in the bone marrow, it goes there, destroy the bone marrow, destroy the bones. But it's actually the hematopoietic problem. So the clinic it's very heterogeneous. Some patient can survive over years, but other patient will survive very short. Maybe just the aggressive can only survive 2 years. And we so far we have a lot of drug. So we try to give all the drug to the patients, but we do not know. So we have no way to predict which patient will benefit from from a standard treatment, which patient actually will not benefit from the treatment. And our goal is like that. So we know that proteasome inhibitor like bortezomib is used worldwide as standard therapy. It's not just in Europe or in America. It's in China. It's also like that. So it's included in the healthcare system as the essential medicine. But in real life, it's only 40 to 50% of multiple myeloma would respond to proteasome inhibitor treatment. So over 50% of the patients, it's a wrong treatment or over treatment. So we aim to develop a diagnostic method to distinguish the responder from non-responder. How we did it? So we form the similar hypothesis. So multiple myeloma actually originates from the plasma cell development. So the plasma development originates from B cells. When the B cell is activated by antigen, it will go through a process in the germinal center. So here you have somatic hypermutation. So to make antibodies, antigen binding site more specific or have a much higher affinity. You also have a class switch. So initially we have IgM. So here we have IgG, IgA, and so on. So we use the regulators of germinal center initiation or formation and do the Pearson coefficient analysis in the glioma dataset. We want to find, or we use bioinformatics to screen for gene networks. We want to find which gene network will allow us to do the similar job in glioma. Actually, so we found out MCL1 co-expression module. So MCL1 is located at the chromosome 1q. So we can see many genes co-expressed with MCL1 is located at 1q. And MCL1 is also essential for plasma cell survival. So we use this MCL1M so we can distinguish myeloma into subtype which have high expression of MCL1M, low expression of MCL1M. No matter where this myeloma is treated, this low group always have better survival, high group always poor survival. It's different. It's always like that. It's in Europe like that, in America like that, probably in China will also be like that. And so this data is from America, and this data is from Europe, actually from phase 3 clinical trial in Holland and Germany. So you see high MCL1M high group, poor prognosis, low group, better prognosis. And the beauty in this they have designed 2 treatment arms. One is called PAD arm, which contains proteasome inhibitor. Another one is VAD arm. It's standard chemotherapy treatment. You will see like that. So if we distinguish myeloma as 2 subtypes, there's a high group and low group. You will see that if we compare the 2 treatment arms within the subtype, only within the high subtype, the PAD treatment improves the PF, improves the survival, improves survival, and in the low group, no improvement of the survival. So the benefit is only seen in the MCL1M high group. And this is so in newly diagnosed patient, and a similar trend we can see that is also in the relapse patient. This is another phase 3 clinical trial called APEX phase 3 clinical trial. You see, this is proteasome inhibitor compared with steroid treatment. The proteasome inhibitor benefit is only seen in high group, not seen in the low group. So with this, I want to almost finish my presentation. You see, so what we should do in the next. So we want to, based on our classification scheme, we want to identify subtype-specific etiology. And we hope that with the understanding of the etiology, we may be able to develop subtype-specific treatment, particularly for glioma. We are also working on other diseases. We are looking for more collaborations to apply our bioinformatics expertise to the other diseases. So at the end, I want to thank my students. They are from Beijing, and also my collaborators. So you see, I myself don't have, I'm not working at the hospital. So the clinical data is contributed by the collaborators and also from a database. And although I have supervised the project, most of details actually are solved by the students. So with this, I thank you for your attention, and we can start to have a discussion of the questions. Thanks.
Okay, thank you very much for the great talk, Dr. Fan. So now we'll move on to the Q&A session. So please be sure to ask any questions you may have for Dr. Fan in the Q&A box of the Zoom interface. So first off, we'll start with why is EM/PM classification so powerful?
Because I think that's so. I just I can't. Can I still go back to the slide? I'm not sure.
I'm not sure.
Yeah, maybe I can't. I can't.
I think you should be able to press backwards.
Really? Sorry. No. Okay. See. Okay. Sorry. I think so. You have different ways to look at the same question. So this is the unbiased approach. So you simply based on the G expression analysis. And what we do is that we want to look at the evolution. So something which is evolutionary causal, which is both important for the organ development and also frequently change in the cancer genome. And this signature will be powerful. Where sometimes with this signature cooperate with cells origin will determine the biological feature of cancer, no matter in glioma or in other cancers. I think this principle is true for most of cancer, most type of cancers.
Okay, so the next question is, why do you think there was such a reluctance to accept the EM/PM classification schemes you proposed, despite the clinical relevance and consistency of the data provided?
Oh, that's a good question. That's a good question. I think I'm actually reading a book of how science is evolving. So people tends to work on dogma, follow the dogma, and the field is happy to defend existing dogma. That's always happens like that. Not just in glioma field, in many fields, I think always in other fields, it's also like that. So new ideas, new concept, it takes time, it requires persistency and courage to reach that bar. A good question.
So we have another question. So in addition to myeloma, can this strategy also be applied for other such diseases? And have you looked into that already?
We have looked very little on this issue. We're looking for microenvironment, tumor microenvironment, and this is we can see very interesting data, extremely interesting. So how tumor microenvironment affects the progression of looks like similar type of cancer. Genetically looks like similar, and serial origin also looks very similar.
So have you and your lab considered implementing deep learning or machine learning to the data and the classification schemes?
We do a lot.
Mm-hmm.
We do a lot. So we actually, but I think the deep learning, machine learning has to be guided by the biology. With this is a typical example of a deep learning, machine learning. Just a moment, I hope I can see it. This is a typical example of what's happening in the field. So if you don't have so I think the evolution and genetic both are important. If you don't have evolution and genetic as guidance, those machine learning is I think those paper or those outcome fail to distinguish what's the real signal, which is noise. Just I say like that. Also like this.
Mm-hmm.
So we see a lot of.
No. So yeah, so the next question is, would you be open to answering more questions after this webinar? And how can we reach out to you with those questions?
You can reach me by email. Yeah. You can see in the maybe there's a blocked by this. Sorry, I'm seldom using Zoom. my email address is under my name actually.
Right, we can see it. Yes.
See it? I can't see it. Sorry.
So I think that's all for the questions we have received at the moment. So yeah, now I'll pass things back over to David to conclude.
Okay.
Thank you very much, Nicholas. And on behalf of Medical Valley Alliance, it's just for me to say see you later. I hope it has been time well spent, and that you have appetite for more Medical Valley Alliance events in the future. And as mentioned earlier, feel free to reach out to me if you want to know more about this and other Medical Valley Alliance event concepts. Finally, I would also very much like to thank Nicholas and Dr. Fan and the whole GenScript team who made this webinar possible. And I guess with that said, I think we have reached our destination, so to speak, and I will conclude this inspirational webinar. Take care out there.
Okay, thanks a lot. Thanks a lot. Okay.